Okay. Good evening. Can you hear me? Yes. Okay. Can you hear me? Yeah. Hello, people. Everyone is online. Yes. So we take the final step in the paper class today. Okay. So we are going to move on to the proper question based, the exact A level question based final revision of the important theory points. Okay. That's what we are going to do, right? It's my belief model questions are no longer useful at this point, right? Model questions are good to practice sort of hidden things in the syllabus that you haven't studied or understood exactly in the theory, but now it is time to practice for the real thing. Okay, time to practice for the real thing, the real exam questions. Okay, so that's what we are going to do. Okay, so let's get the start into this. So we are going to start this past paper question discussions from the 2021 paper. Okay, so because 2019 2020 was a good paper, 2019 didn't have applied biology and some of the question patterns were kind of different. So that's why I decided to start from 2021. Okay, so let's see. So I believe, let's say, just to keep it interesting, how about we start from the animal form questions? How about we start from the animal form questions that we have in this paper, and then I'll explain the theory around them, and we can go ahead. Okay, with we'll come back to plan form. Okay, we'll come back to plan form, and so in the revision class we have a 2-day applied biology session happening. So we have finished part 1 of the applied biology. So day 1 applied biology we have already finished, and we have just one more day and applied biology will be over. Okay, then I'm going to have a day dedicated to molecular biology revision. I'm going to have a day dedicated to molecular biology revision. Okay, then that is going to be the end of the revision class. Okay, so that's then there's another final day left in this month, and that is going to be the end of the revision class, the revision program that we had. Understood? Okay, then we are going to have further question discussions, past paper discussions, and then so you can go to the exam. Okay, just keep it simple, keep it stress free. That is the main thing, because these last weeks days are there for you to collect your thoughts, collect your knowledge, sort of gather everything into one place, gather all the knowledge that you have gathered, organize that, right? Always තැන් වල තියෙන information ටික organize කරගන්න, then take that information to the exam. This is the time to do that. Okay. Okay, we'll start from question number 18 onwards. You can ask any question if you want further clarification or whatever. Okay, which of the following tissue location combination is correct regarding the human body? So we are going to go ahead like reviews, writing reviews per each question. Okay, can you hear me? It's raining a little bit. Okay, can you hear me? It's all clear, right? Okay. Let's see. Loose connective tissue found in the tendon. Is it correct? Right. So if you correctly match what is found in the tendon, a tendon is where a muscle connects to a bone. A muscle connects to a bone by a tendon. So it requires a lot of strength. So are you going to be able to do that with a loose connective tissue? No. You are going to require what? A dense connective tissue, or we can call it a fibrous connective tissue. And you are going to require a dense or a fibrous connective tissue. Okay, that's what you are going to need in the tendon. So that's false. Adipose tissue lining of the mouth. Which epithelial? Okay, which tissue do you have in the lining of the mouth? Yeah, lining of the mouth. It's an epithelium. It's a lining, no? So it's an outer surface. What is the definition of an epithelial tissue? Lining inner or outer epithelial, free surfaces, right? So lining of the mouth is going to have some sort of a epithelium. What epithelium is it going to be? What is it going to be, the epithelium? Yes, it's a stratified squamous. It's a compound epithelium. It's a stratified squamous epithelium. Why is it a stratified squamous epithelium? Because it's skin, no? It is covered by skin, but you are going to need multiple layers of keratinized cells, sorry, non-keratinized cells, I'm sorry, multiple layers of squamous cells in order to protect the lining of the mouth from all the rough food that you eat, right? All the rough food, dry biscuits, dry bread, right? Dry cookies, or you know, you can feel your mouth getting chips, right? Whatever chips you can feel your mouth getting sort of damaged, but it is going to be, you are not going to bleed because you have several layers, no? So your mouth lining is stratified squamous, but is it keratinized? No, it's not rough as your palms, ne? So it's stratified squamous. Then anus. Yes, your anus has, because of the defecation is a friction, it's a process that is producing friction, because the feces can be sort of semi-solid, okay? It can be constipated dry feces as well. So the anus has stratified squamous. Can anyone tell me what are the other places you can have stratified squamous? Vagina. Those are non-keratinized, have the non-keratinized stratified. Anus. Okay, vagina is of course non-keratinized, otherwise the sexual intercourse is going to be very painful. Okay, got it. Then intestine simple cuboidal epithelium. Which epithelium are you going to have in the intestine? Intestine should be modified for absorption. So what is the epithelium that is designed to do absorption? Is කියන්නේ it's columnar නේද? This columnar epithelium. You can write in front. If you have a paper with you, you can write in front. It is columnar epithelium. And this columnar epithelium is what? Having what in the digestive system? Has your microvilli, නේද? Microvilli. So microvilli also increases surface area. Pseudostratified epithelium in the kidney tubules. Pseudostratified epithelium, we can say pseudostratified epithelium is particularly found in the respiratory system. Respiratory tubes. Okay, respiratory system, sort of the conducting system. Kidney tubules has what? Kidney tubules are designed. So if you have like a nephron and the sort of the distal convoluted tubule and the proximal convoluted tubule, what is it designed to do? So we are saying we are asking what cells are found in the wall of this nephron. What cells are found in the wall of this nephron? So this nephron wall is designed to do what? It is designed to secrete things out of the urine that did not get filtered in the glomerulus into the blood capillary network that is surrounding it. No? So for secretion, what is the epithelium designed for it? It is cuboidal. No? Simple cuboidal epithelium that is designed to do this. Okay, got it? Right. 19. In which of the following will the release of one result in the stimulation of two? So you have to relate number one versus number two. Okay. So, okay, you have gastrine, ABC, gastrine, cholecystokinin, and secretin. And then what do you have on this side? Production of gastric juice, secretion of gastric juice, release of bicarbonate from the pancreas. Okay. What do you think? One results in the stimulation of two. Gastrine, what is the action of gastrine? Give me 1 minute. Okay. Hello. Oh. Okay, can you give me 5 minutes? I'll be back soon. Okay. Take a small, I'll be back within 5 minutes. Okay. Just hold on. Okay, right. Let's- Okay, right. Let's continue. All right. Okay, where were we? So number 19. So you have gastrin, cholecystokinin, and secretin. They're asking which one stimulates number two. Yeah. What do you think is the effect of gastrin? What do you think is the effect of gastrin? Is it something related to the production of gastric juice or something that is doing what? Secretion of gastric juice. What do you think? Gastrin. Yeah. Any idea? So keep the discussion a bit active. So what do you think? Gastrin hormone? Yeah. Actually, the production is under the control of the gastrin hormone. Okay. The secretion of gastric juice is under what sort of control? The secretion or the release of gastric juice is yes, it is this is the production. The release of gastric juice is under what? Yes, what control? It is under nervous control. Okay, do you understand? It is under nervous control. When food arrives in the stomach is stretched. The stomach is stretched. The stomach wall is stretched. So whatever gastrin that was, sorry, whatever gastric juice that was produced is stored in the gastric gland and it will be released when food arrives. Okay, so it is kind of a nervous reflex, the release, but the production is under the endocrine control of the gastrin hormone. Okay. Then the next, cholecystokinin and secretin. What is the effect of cholecystokinin? Cholecystokinin doesn't have anything to do with secretion of gastric juice. Cholecystokinin, what does it do? It triggers the release of bile. You can write here, cholecystokinin triggers the release of bile from the gallbladder and also digestive enzymes. Yeah, digestive enzymes. From where? From the pancreas, pancreatic digestive enzymes. Secretin has a completely unique function. Yes, it releases simultaneous release of bicarbonate. So if you take pancreatic juice, right? If you take pancreatic juice, what are the two components of pancreatic juice? Yes, you of course have pancreatic enzymes, and also your pancreas has the ability to produce bicarbonate ions. Why do you need the ability to produce bicarbonate ions? That is to neutralize the acidic chyme that is coming from the stomach. Okay, so secretin controls bicarbonate. So the answer is A and C. No? The answer is most likely to be A and C. Okay. 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 Later I'll explain this again with my with the diagrams. Okay. Yeah. Shall we move on? Okay. Right. Then, question number 20. Which of the following is most likely to happen if the tricuspid valve does not close properly? So, Taggala, you need to have a sort of a visual idea, visual imagination of the arrangement of the heart. So, you have the two atria, the two ventricles separated by the interventricular septum and the interatrial septum. Which side do you have the tricuspid valve with the three cusps? Which side do you have the tricuspid valve and which side do you have the bicuspid valve? The tricuspid is found on the right side, no? Right atrium, right ventricle. Bicuspid is found in the left side, left atrium. Okay, got it? So, tricuspid valve does not close properly. Right atrium will not completely into empty during atrial systole. What do you think? Is this correct? Atrial systole means, yes, the atrial contraction. Systole means contraction. So when atrial systole happens, some of the blood, I mean, the blood from the right atrium will go into the right ventricle. So, if the valve is not closing properly, is it going to affect the blood from right atrium to ventricle in that direction? The downward flow, is it going to be affected? No, right? The atrium will completely empty. No problem with that. Atrial systole is not going to be affected if the valve is defective. Left atrium, completely irrelevant. Either completely irrelevant. Left atrium is not in the picture. Amount of blood that flows into the right atrium will be reduced. Again, nothing to do with the left, sorry, tricuspid valve closing. Amount of blood that flows into the lungs will be reduced. Why do you, is that something? So, I mean, can you consider that statement? What do you think? Yeah. So from the right ventricle, what is the blood vessel that is taking? I mean, what is the blood vessel that is starting? The main artery that is starting from the right ventricle. You have the pulmonary artery, right? Pulmonary artery starting with the semilunar valve here, starting with the semilunar valve in the right ventricle. So when the right ventricle contracts, blood should go into the pulmonary artery and go into the two lungs. But when this tricuspid valve is does not closing properly. අපි ඒකට කියනවා regurgitation එකක් කියලා. You don't need that. Regurgitation කියන්නේ උඩට එනවා කියන එක, right? You are food regurgitation means කාටට එනවා ආපහු food, right? What's going to happen is this blood is going to go back into the right atrium without going into the pulmonary artery. Okay? Therefore, blood flows into the lungs will be reduced. Yes. When the tricuspid valve is not closing properly. Can you understand? Some amount of blood will flow into the left atrium. That is not relevant because tricuspid valve is found in the right side of the heart. So can you understand? You need a very nice overall idea of the structure of the heart in order to answer this. Okay, you need to remember the which side of the heart you have the valves and also different blood vessels that are connected. Okay, clear? Okay, 21. Which of the following is caused by, so 2 is caused by 1 during homeostatic control of breathing. So we are going to a bit of respiration. 1 caused by 2. Blood pH decreases. Okay, blood pH decreases. That means what is the cause of blood pH decreasing? CO2 dissolving, excessive CO2 in your blood dissolving in water and forming carbonic acid. That carbonic acid is dissociating into bicarbonate and H plus. This H plus is going to decrease the pH as blood becomes more acidic. Then, so caused by 1, I'm sorry, caused by 1, okay, 2 is caused by 1. When the carbon dioxide level in tissue increases, yes, blood pH decreases. That is correct, no? Yes. 2, medulla oblongata detects decreasing pH of cerebrospinal fluid. Yes, your medulla oblongata, if this is the brain, okay, this is the starting of the brain stem, okay, so you have the midbrain here, midbrain, then pons and the medulla, no? So medulla is part of the central nervous system that is extending to become the spinal cord. This whole thing, this our brain is completely bathed in not blood, right? The nutrient supply and the removal of waste through the brain is provided by a special fluid that is cerebrospinal fluid, no? So it is found inside the brain as well. In where? The brain ventricles. So the CSF is found in contact with the medulla. So medulla is able to detect the changes in pH of cerebrospinal fluid. That is correct. Okay. So it detects the decreasing pH because from the blood, carbon dioxide can go into the CSF. Okay. So now the medulla is detecting, okay, your carbon dioxide level has increased. Then what should the medulla do? It should do something to get rid of that carbon dioxide. That is done by breathing fast and hard. So you have to breathe deeply. not decrease. So you have to increase the depth. So this is false. Yeah. C. Sensors in the aorta detects high concentration of carbon dioxide in blood. Medulla oblongata receives signals from aorta. Be very, very careful. So if you studied halfway, bagainam padam kare, you won't be able to answer this. Why? Because, okay, you might remember, okay, aorta plays a role in maintaining homeostasis, okay. Then maybe if you didn't read a question, you are going to put it correct. But sensors in the aorta, yes, there are sensors in the aorta, but are they detecting carbon dioxide concentration? No, they are detecting again the pH of blood, not CO2, because CO2 is going to force increase the H plus. So it is not detecting the carbon dioxide, but it is detecting the pH. So you can write in front, detects the decrease in pH. Then the aortic sensors will send signals to the medulla to increase the rate and depth of respiration. That is the correct way. So answer number C is also, sorry, C is also false. So you can take only answer number 1. Can you understand the tricky things in this? Because the aortic detect sensors detect the pH, not CO2 concentration directly. The CO2 concentration is not directly detected. Okay, question number 22. B lymphocytes. Okay, so you know you have two types of lymphocytes as T and B lymphocytes. Completes the development in thymus. So both T and B lymphocytes are formed where? In bone marrow. Then some will go into a further training camp. That further training camp is called the thymus. Some will remain in the bone marrow itself and they will be B lymphocytes. The people who go to the thymus to complete their training, it's like to or at the university to complete your education. Those are going to be called as T lymphocytes. Okay, so B lymphocytes do not go to the thymus. Okay, are mainly responsible for cell mediated immunity. Who is responsible for cell mediated immunity? It is T lymphocytes. So remember, T lymphocytes are the cells that are capable of killing other cells. T lymphocytes can kill other cells, therefore, cell mediated immunity. Cells killing cells. Okay. B lymphocytes do not directly go and kill cells. They release a specific protein that is called antibodies. They will have a sort of a neutralizing action and so many other things. Okay, so you can write it front. It is not B lymphocytes, T lymphocytes. They are not involved in naturally acquired active immunity. They are. Okay, once you get a certain pathogen attack, could be a virus or a bacteria, there could be memory B cells producing, and those memory B cells will remain for a long time and they will give you protection for subsequent. Again, IR, if you get the same infection, you will be protected. So you can't say they are not involved. Can differentiate into natural killer cells and helper cells. This happens to what? It is again T cells. T cells has what we call as two effector forms. T cells has what we call as two effector forms. Okay, so whenever you are discussing these questions with me, I want you to take down small points that is going to help you study these things for the final time. So tonight itself, go and study the things. Okay, if you feel like okay, but matakana exactly how T lymphocytes and B lymphocytes do cell mediated or antibody mediated immunity. So tonight itself, go and study. Make a list of things that you can't remember. So there are two effector T cells forms, that is the T cytotoxic T cells and helper T cells. Cytotoxic T cells are like they have guns. They can kill other cells. Okay, they can, using specific chemicals, they can kill other cells such as virus infected cells or cancer cells. Okay, contain antigen receptors on the plasma membrane. Yes, that is common for both T cells and B cells actually, because that's how they get what? What do you call when an immune cell gets activated? What is a special name that you give to call an immune cell that is activated when they detect there's something foreign. Could be a pathogen. What do you call that when an immune cell is activated? Anyone? Sensitization, no? Sensitized. Sensitization, no? They become sensitized. So answer is answer number 5. Understood? 23. Excretory structures of crustaceans, annelids, and flatworms are respectively. So you have crustaceans. Crustaceans are a type of arthropods. Okay, so they come under arthropoda. Annelids. So annelids. Then you have platyhelminthes. What are their excretory structures? What are their excretory structures? Yes. If you take in the order of evolution, who are the first organisms to develop a specialized excretory system? Who are the first organisms to develop a specialized excretory system out of these? So evolutionarily simple mechana. Who is the most evolutionary simple one? It is the flatworm. Ne? The evolutionary most simple one is the flatworm. So flatworms, what did they develop? Yes, flatworms, what did they develop? They develop unicellular excretory structures. Ne? Just uh, excretory cells. They just develop some excretory cells. What are those excretory cells called? Flame cells. Yeah, they are called flame cells. So the final answer should be flame cells. Flame cells. There are two answers. Okay, flatworms. Then annelids. Next came annelids. Annelids started to have what sort of excretory system? They started to have a multicellular one, and that is opening to the outside. Okay, so flame cells are single cells that are involving excretion. Then annelids, as the next step, they started to have multicellular structures. That is, they were called as nephridia. So you have an answer as nephridia. Okay. Then finally, crustaceans. Crustaceans are unique for having what? Green glands. Okay, that's an MCQ point you need to remember. Green glands. They are mainly for their sort of salt excretion. Okay, usually located on the head. So you can see in crustaceans such as your prawns, right? You can find the green glands. So answer number is 5. Clear? Okay, 24. Correct statement regarding human brain. So we are moving on to a bit of nervous system. Human brain stem is developed from the embryonic midbrain and hindbrain. Okay, I can quickly show you something that is going to help you revise this, sort of remember this. The brain development. Okay, brain development is an important concept that you need to understand. Sorry, not this one. Brain development from the embryonic brain, right? From the embryonic brain. So your embryonic brain is having 3 parts as the forebrain, midbrain, and the hindbrain. Forebrain, midbrain, and the hindbrain. Okay. So you need to remember embryologically which part of the mature brain or this fully grown brain came from which parts in the embryo. So can you see this is embryo at 1 month. Okay, which parts came from which parts you need to remember. So from the forebrain, what came? What came from the forebrain? The cerebrum, thalamus, and the hypothalamus. Okay, so from the forebrain you got the cerebrum. Okay, the thalamus is sort of located here, and of course this is the hypothalamus, and also the sort of the thing that is hanging here, that is pituitary. Okay, and you have hypothalamus. Remember them padanka neither from the forebrain. What did you get? Cerebrum, thalamus, hypothalamus, and of course the pineal body. The midbrain is actually going to remain as the midbrain, which is part of the brain stem. Okay, what are the three parts of the brain stem? You have midbrain, pons, and medulla oblongata. Okay, hindbrain gives rise to what? Three things. The hindbrain gives rise to, yeah, hindbrain gives rise to pons. Sorry, so you can start with cerebellum, pons, and medulla. So who is cerebellum? The sort of the part of the brain that is below your occipital lobe. Yes, cerebellum, pons, and medulla comes from the hindbrain. Okay, this is the final revision. Then okay. Okay, let's move on back to the paper. Okay, clear? That is just the diagram at 5 weeks, right? Jump to this. Okay. Brain stem is developed from the embryonic midbrain and hindbrain. What do you think about that? Yes, what do you think about that? Brainstem. What are the three parts of the brainstem? You have midbrain, pons, and medulla. So midbrain comes from, of course, the midbrain. Pons and medulla comes from the hindbrain. So that is correct, no? So the brainstem. These three together is called as the brainstem. These three together, midbrain, pons, and medulla is the brainstem. But the midbrain comes from the midbrain, pons and medulla from the hindbrain. Very correct. Frontal lobes of the cerebral cortex contain the visual sensory area? No. Your vision is actually sensed by the occipital lobe at the back. Okay. So we have seen some people who have struck their head. They have struck their head at the back of a maybe chair or a side of the bed or maybe a short wall when they are falling back. If they have struck their occipital lobe at the back of the head and there's some damage, some bleeding, they lose their vision, pain, their eyes are completely okay, right? Their eyes, no damage to the eyes, but they say, okay, I can't see anything. That means the visual cortex, visual sensory area has been damaged. Okay, so you can correct this. Where do you have? In the occipital. In the occipital lobe, you have the visual sensory area. The midbrain contains the fourth ventricle. Where do you have the fourth ventricle? You can tell me by looking at this diagram. Brain ventricles. These are the brain ventricles. So, how many ventricles do you have? How many ventricles do you have in the brain? Yes. How many ventricles do you have? You have four ventricles. You can see Metanadino dekak, right? So you can see left and right killer. There are two ventricles here. This is called the third ventricle, and this is the small one that is here is called the fourth ventricle. Okay, so you can see Medekai, and this third one is located where? In the top part, sort of mostly located in the cerebrum. Yeah, cerebrum area. So you can say the first three ventricles are located in the forebrain, and the fourth ventricle is located. It is actually sort of behind pons, the fourth ventricle. Okay, so it is located in the hindbrain. Clear? Come back to the paper. Midbrain contains the fourth ventricle? No, you can correct this. Where do you have the fourth ventricle? Hindbrain. Corpus callosum connects the two hemispheres of the cerebellum? No, it is connecting the cerebrum. It's like karme. It's like a sort of a bridge of wires, the bridge of nerve fibers that are connecting the two cerebral hemispheres. Okay, because they should work together. No? Even though you have two hemispheres, they should work together. Okay, so they connect the two hemispheres of the cerebrum. Thalamus regulates sleep and wake cycles. No, thalamus is like Arame. In a computer, you have like a to process all the inputs. You have a sort of a processor. It's like that. The thalamus contains sort of receives all the inputs, sensory inputs. Okay, but things like sleep and wake cycles, sexual habits, and the hunger, satiety, thirst, those autonomic responses, fight and flight, or auto they are all under the control of hypothalamus. In the hypothalamus, you have nucleus for everything. Or hunger, thirst, everything. You have nucleus. It's the hypothalamus is a ball of nuclei. Okay. So what's the answer? Answer number one is 24. Okay, 25. Events that occur after the membrane potential of a neuron is changed above the threshold value are given below. Okay. So what do you think? So you have a membrane, a neuron that is resting. I can revise this quickly like this. Okay, listen. What do you mean by membrane, membrane that is resting? A resting membrane is something like this. A membrane or nervous a neuronal membrane that is resting is something like this. So you can say inside you can see this orange color squares, that is the potassium ions K plus. Okay, I'll write here K plus. So inside the cell you have a higher K plus concentration inside the nerve cell. Okay, then outside you can see there's this yellow color balls, those are sodium ions. Sodium ion concentration is higher outside. Okay, so that is how a resting potential is. And also you need to know the inside is more negative compared to the outside because of what? So you have large proteins which are usually negatively charged. Okay, you have large proteins. So while I'm doing this, you have to turn to the resource book at that page. Okay, go through the resource book wherever the question is. We are going in that order, innit? Okay, we are in nervous system now. Okay, so because of these certain large proteins, the inside is negatively charged. Okay, so this is resting, the non-conducting membrane. Whenever you have a nerve impulse or a signal that is enough to generate what we call as an action potential. Okay, so you know you have the graph now. Yes, you have the graph at around minus 70, the membrane is resting. Okay, if you get a signal that is kind of close to maybe this value, okay, that is called, you have a certain special value, that is this value that I call as the threshold value. If there is a signal that is enough to cause the membrane to reach the threshold value, what's going to happen? Yes, can. What's going to happen is there are what? There are voltage gated sodium channels. Voltage gated sodium channels are there. Through the voltage gated sodium channels, there's going to be a massive inflow of sodium into the cell. Therefore, the cell is going to get depolarized. Then after some time, the sodium channels are going to close and the potassium channels are going to open so that the potassium goes out. Okay, then the cell membrane potential starts to come back to the normal level. It goes below the resting and then come back to the resting. Okay, this is the graph now. So this is depolarization. On this side you have repolarization. So at this point, don't think about complex things. So what you need to know only, when you are depolarizing, you have what? Sodium influx. Sodium ඇතුළට එනවා කඩාගෙන. When you are repolarizing, potassium efflux. Potassium is going out. Okay, This is depolarization or the generation of an action potential. Understood? This is what happens. Okay, shall we come back to the question? Events that occur when the membrane potential is reached above the threshold level. So you have something, okay, maybe loud sound, okay, maybe a touch, something had the in energy more than the threshold value. So what is going to happen? You are going to generate a nerve signal. What's going to happen? What is the order? What should happen first? Yeah, sodium inflow. Sodium channels open, sodium comes in. Okay, that is going to cause what? When sodium comes in, the inside of the membrane becomes more positive. So we call that the membrane becomes depolarized. Okay, so one is sodium coming in, two membrane become depolarized. Then what's going to happen? Sodium channels are going to slowly close, potassium channels are going to slowly open, causing a potassium outflow, so that the inside becomes gradually more and more negative again. Then you can say the membrane is coming back to the normal resting level. So you can say BDAC. Answer number 3. Yes. Can you understand? Keep it like a story, right? Keep in your mind like a story. Do not overcomplicate things. Very complicate කරගන්න යන්න එපා. Step wise මතක තියාගන්න at this point. Okay. All right. We are moving on to a bit of endocrine. Okay. Bit of endocrine. Let's see. So hormone effects. Okay, hormone and its main function. So adrenaline, is it long term? General knowledge, again, you can answer this. Yes. Do you get adrenaline for a long time? No. It's a very short burst of adrenaline, short secretion. So you can write in front, adrenaline is a short term stress hormone, short term stress hormone. Can anyone tell me it is secreted by what part of adrenal gland? It's a adrenal inner medulla. Inner medulla secretes adrenaline. Prolactin, okay, something to do with the milk. Is it milk ejection? Read very carefully. Is it milk ejection? What do you mean by ejection? Releasing, squirting out. Well, you are releasing. Is prolactin related to milk release? No. Who is going to do the milk releasing part? There is another hormone that is called as oxytocin. Oxytocin is released when the baby is suckling on the mother's breast. Prolactin is not related to milk ejection. He is the one milk production. Prolactin stimulates milk production. Melatonin regulates innate immunity. There is nothing related to immune regulation by melatonin. It sort of sets your daily rhythm, your sleep and wake cycles. Yeah, your daily rhythm. අපි කියනවා diurnal rhythm එක කියලා. Daily rhythm. When do you go to sleep? When do you feel sleepy? When do you wake up? ඒ වගේ. Thyroxine increases metabolic rate. Yes. People who had excessive thyroxine, hyperthyroidism, they know. Okay. Sweaty, weight loss, sweaty skin, heart beating fast, high heart rate, those kind of things are indications of high metabolic rate. Weight loss, right? Your engine is running faster. Thyroxine. Luteinizing hormone or LH. Who is the hormone involved in stimulating sperm production, stimulating sperm production? Spermatogenesis. Who is the hormone involved in spermatogenesis? Sort of stimulating spermatogenesis. Yes, what do you think? Yeah, LH. LH or FSH? What do you think? Can. So, LH is not having a direct effect on the sperm production. Yeah, it is FSH, right? Because imagine, so what I tell you to remember, okay, the name says follicle stimulating hormone. So, if you take the female system, what is it going to do? It is stimulating the follicles to develop and later to produce the egg. Ovum is produced at the end of the follicle maturation. So, FSH matures the follicles. So similarly in the male, what is the similarity? What is the similar thing in the male? That is sperm, no? So FSH in the female helps the production of egg. Then FSH in the male helps the production of sperm. Okay, FSH. So spermatogenesis is stimulated directly by FSH. What is the effect of LH? Can you write down? LH stimulates a type of cell that we have in the testes. Can anyone tell me? LH, mataka lazy. LH for L, Leydig cells. Yes, Leydig cells produces testosterone. So, of course, testosterone has an indirect effect on spermatogenesis, but directly it is FSH. Okay. Any questions? Shall we move to 27? Okay. Going too fast. All right. Do you want more clarifications? Okay, we are moving on to a bit of reproduction. I mean, already there. Okay, after endocrine. Okay, right. Women, meiotic division of the secondary oocyte released in ovulation is arrested. Okay, so oogenesis, bit of a complicated thing. So, what's going to happen? Meiotic division of the secondary oocyte released in ovulation is arrested at what phase? At what phase? What do you think? අපි කිව්වට, so if you can understand this, even though we say a female releases an egg during ovulation, does she actually release an egg? Does she release an egg really? No. She releases a secondary oocyte. She only releases a secondary oocyte. A secondary oocyte is going to divide to be the mature ovum and the second polar body. So even though we say during ovulation, and so it seems like there's an egg coming out, but it's not the egg. The ovum will be produced only in what situation? If there is a sperm, if there is a sperm penetrating this secondary oocyte, only then this secondary oocyte will complete meiosis. What? Which phase of meiosis 2? Yes. So actually, meiosis 1 is completed when you are producing the secondary oocyte. So you have primary oocytes, no? Primary oocyte is going to divide by meiosis 1 to produce the secondary oocyte, and what else? First polar body. Okay, first polar body. So when the baby girl is I mean, when she is born, she already has all the primary oocytes she is going to need in her life cycle. She already has all the primary oocytes she is going to need. Okay, so those primary oocytes are arrested. I mean, they actually started the meiosis, but it is arrested in prophase, so nothing has happened, right? Meiosis started. Then, after puberty, what's going to happen? Meiosis 1 will be completed. Sorry, so it's meiosis 1 here, and they will start to produce secondary oocytes and first polar bodies. Secondary oocytes are released every month, almost every month, every 28 days, whatever. Then, if there is a sperm penetrating this, then only it will become a oocyte. There's a question. If the egg is not fertilized, what will happen? They just pass out. Every ovulation or every period that is not ending up in pregnancy, all the oocytes are just going out. Yeah? In any period that is not ending up in pregnancy, they are just going out. Okay, you don't even see they are very microscopic, no, not like a chicken's egg, no, not very, they are very small. Okay, so which phase? Okay, where are we? Oh, in secondary oocyte. So the secondary oocyte is arrested in metaphase 2 actually. So you can actually see, if you zoom into the secondary oocyte, you can see the chromosomes are arranged on the metaphase plate. You can see the chromosomes are arranged on the metaphase plate, and you can actually see these microtubules, kinetochore microtubules that are connecting to those chromosomes. So it is actually metaphase 2. That is uh, the state of the oocyte. But clear? Do you want me to explain oogenesis? Do you want me to explain oogenesis? All right. Then let's move on to human development. Okay. So we are talking about amnion here. What is amnion? Yeah. What is amnion? Amniotic sac. Right. Amnion forms what we call as the amniotic sac that is around the baby. Right. Amniotic sac is filled with what? These are points. Amniotic sac is filled with what? Amniotic fluid. Right? Amniotic fluid is mainly composed of baby's urine. Okay, baby, the blood that the urine that is the baby is producing. Okay, so you were in your own urine for a long time. Okay, so anyway, so the amnion produces the amniotic sac. Okay, so what are the functions of the amnion? It entirely surrounds the embryo. Okay, sort of providing a bit of acting as a bit of a shock absorber and those things as well. Okay, so acting as shock absorber and prevents desiccation. What are the other functions? Who? Okay, there are four embryonic membranes. No? There are four embryonic membranes. What are the four embryonic membranes? You have chorion, amnion, allantois, and yolk sac. What are the individual functions? Let's see if we can match them. Who is going to produce HCG? HCG hormone. What is the function of HCG hormone? HCG hormone is going to keep the corpus luteum longer. Corpus luteum is going to be maintained by HCG. Why do you need the corpus luteum? Because corpus luteum secretes estrogen and progesterone. Estrogen and progesterone maintains the uterus lining or the endometrium. So what will happen if you don't maintain that? The small embryo that is very small, it's going to go away with the endometrium. What do you call the breakdown of the endometrium? Period. So if you don't have someone to keep the corpus luteum, the woman is going to get a period, menstruation. Then the endometrium is going to go away together with the chooty baby, right? So as soon as the embryo is implanted, it is going to start producing a hormone that is called as HCG. Okay, HCG will later be produced by who? The chorion. Chorion is actually developed from the blastocyst, the trophoblast of the blastocyst. Fetal portion of the placenta. Fetal portion of the placenta is what? What is the main fetal portion of the placenta? Again, babage පැත්තේ තියෙන්නේ, that is also chorion. The baby's side of the placenta is formed by the chorion. Mother's immune responses, who does that? That you can say the entire placenta, the entire placenta. But if yeah, it is given as chorion, yeah, better take it as chorion. Entirely surrounds the embryo and prevents, method you can write down, protects from desiccation and act as a shock absorber. So the breaking of the amnion we consider as the first sign that the baby is about to be born. Up here again, water bag break. Yeah. So placenta produces HCG. What are the two parts of the placenta? Right. So maker, you need to understand. You have placenta. Okay. Two parts. Placenta again, it's like a arame. It's like a country, two borders of a country. So one part of the placenta belongs to the mother, one part of the placenta belongs to the baby. Okay, right? The placenta develops one part from the baby, fetal portion, that is called the chorion. Then there's the maternal or the mother's portion of the placenta that is coming from the mother's endometrium, that is the inner uterine lining, the uterus innermost lining. So placenta is a two-part organ. One part belongs to the baby, one part belongs to the mother. Okay, so the chorion of the placenta that is belonging to the baby produces HCG. Understood? 5 serves as the source of primordial germ cells in the developing gonads of the fetus. So the developing gonads actually come from the yolk sac. Yolk sac. Okay, I can show you these things. I think I have Taggalega, I'll show you in the reproduction. Yes. Okay, think of this as the final revision. Okay. Where were we? Okay. So can you see? This is how this stage of embryonic membranes look like. So this is the chorion. Chorion belongs, so this is the placenta. The placenta has two parts. Can you see? One part of the placenta that is sort of maybe one. This is the same as the mother's endometrium. So one part, this sort of light red color part of the placenta is from the mother's endometrium. This dark red color part is called the chorion. That is from the fetal. It is of fetal origin. Okay. So both together form the placenta. Then also you can see the yolk sac that is going to become your gonads. And so the baby, you don't have the amniotic cavity yet. Method. In this diagram, you can see the amniotic cavity. So this is filled with amniotic fluid. Now you can see a nice mature placenta. This is the chorion, this is the maternal side, that is the endometrium. Understood? What else can I show you? That's it. This stage also you need to know. This is the blastocyst stage, right? So blastocyst stage, you know, this is the mother's endometrium. No, this white color area is the mother's endometrium. So your blastocyst has two parts. You have two parts as the inner cell mass and the trophoblast. Okay, inner cell mass as the trophoblast. Inner cell mass actually becomes becomes the baby. The trophoblast becomes the chorion, then later part of the placenta. This is another, yeah, blastocyst, several stages. This is the implantation. This is the implantation happening on which day? usually day 7. Okay, day 7 after fertilization. Okay. Okay, okay. We took a long time to explain that. Okay, because I think these are things that have not been touched very nicely. Okay, 29. After birth, the anteriorly convex curvatures are developed in the lumbar. Oh, sorry, your vertebral curvatures. Okay, what are your vertebral? So jump to turn the resource book into support and movement. Where do you have this? At the end of the axial skeleton, as I can remember. What are the types of curvatures that you have in your body, in your vertebral column? When you were a baby, when you were born, you had only one curvature. What sort of a curvature was it? It was a concave anteriorly. Israim Baluham Babage, the baby is curved like this. It is concave anteriorly. You had only one curve. Then you slowly started to develop another curve that is convex anteriorly. What do you call that curve? That is the cervical curvature. C, cervical curvature. Then later you started to stand. Okay, sorry, not stand, sit. So that is due to another curvature. So what are the four curvatures that you had developed? You have cervical, thoracic, lumbar, sacral. So what is the direction of the primary curvature? It should be concave anteriorly. Israel Beluham, it is concave. So convex, anteriorly convex curvatures, where can you find? You can find in your neck area, cervical, and where else? Lumbar. Convex anteriorly. Okay, curvatures. So you started to, as a baby, you started to raise your neck due to the development of cervical. You started to sit down. Our body you can sit down because of the development of the lumbar curvature. Okay. Any questions? Right. Let's move on. Question number 30. Correct statement regarding human skeleton. Okay. Articulation of axis with the occipital bone permits nodding movements. What do you mean by nodding? So you need to have several understandings for this. What do you mean by nodding first? Back and forth. In order to nod, what are the two bones you need? You need the occipital bone, sorry, occipital condyles at the bottom of the occipital bone. Okay. So yes, occipital bone is involved. But who is it nodding with? Who is he nodding with? So this is, if this is the bottom of the skull, the bottom of the skull is resting on what bone? On two. So this sort of the atlas vertebrae, that is sort of immediately below the occipital condyles of the occipital bone. Then there's the axis bone that is sort of projecting into the uh, atlas. So that atlas and axis joint allows what? Side to side movement. Nodding is between occipital and atlas. Then side to side is between atlas and axis. So you can correct this. Neither you can cut this and correct. Permits articulation between atlas and occipital bone. All carpal bones in the upper limb contribute to form the wrist joint. The wrist joint is a gliding joint, Mehema, right? How is it formed? Between what and what bones? How is it formed? Between which bones? The wrist joint is formed between what bones? Anyone? How do you define? I mean, what are the ways that you can uh, call the carpal bones? How many carpal bones do you have, first of all? How many carpal bones do you have? You have 8 carpal bones. How are those carpal bones arranged? In two rows. Okay, in two rows. You have a proximal row that is closer to your body. You have a proximal row and a distal row. So actually, your wrist joint is found between the proximal row of carpal bones. Okay, I'll show you. Is be found between the proximal row of carpal bones. Okay, proximal row of carpal bones and what? The mainly the radius. Okay, mainly the radius. So I can show you. Can you see? Now this is a nice view of the wrist. Okay, can you see? Can you count the 8 carpal bones? 1, 2, 3, 4, 5, 6, 7. There's a floating one at the other side. Okay. Yes, medine hidden එක. Okay, can you see? 8 carpal bones. 1, 2, 3, 4, 5, 6, 7, 7. And where's the pisiform? Pisiform is this one, maybe. Okay, 8. Okay, those are the 8 carpal bones. So how are they arranged? You can arrange the 8 carpal bones as a proximal row. Proximal row is the row the closest to your body. Okay, so this is the proximal row, maybe. So the proximal row together with the radius mainly forms the wrist joint. Okay. So these are hidden theory points. Okay. All carpal bones. No, not all carpal bones. You can say proximal row. Proximal row of carpal bones contribute to form the wrist joint. Proximal row of carpal bones contribute to form the wrist joint. Actually, 3 proximal carpal bones and the radius. So remember that. Okay, you saw that, right? 3 proximal carpal bones. Then we move to some diseases. Osteoarthritis is a condition associated with the reduction in bone density. No, arthritis is due to the wearing out of the bone. What is related to bone density? Porosis. Osteoporosis. The bones become like a sponge, pores. Okay. Patella articulates with the lower end of the femur. Patella is the knee, sort of the your kneecap. Articulates with the lower end of the femur. What do you think? Yes, femur is your lower leg bone. So your femur is connecting to the radius, and there's the I'm sorry, the femur is connecting to the tibia. Okay, like this, and the tibia. There's the fibula on the other side, and in front of this we have the patella. Okay. So that is correct. Maxilla is the only movable bone in the skull. Maxilla. Can you move your maxilla? Where is the maxilla? Can you move the maxilla? No. This is the maxillary bone, no? Maxilla, the upper jaw. Can you move it? No. What can you only move? Mandible. You can correct. Mandible is the only movable bone in the skull. Clear? Okay. Okay, tell me which questions do you want to do next? Animal form, all done. So you can see we had about 10 questions here, animal form. Yeah, out of 50, 10 questions were directly from animal form. There's probably from question number 40 to 50. What are the animal form questions? Roots, blood circulation. Okay, right. 43 karo. Okay, plant, please. Okay, yeah, I'm going to do plant next. Yes. So, did you do the this paper beforehand, or are you doing it right now with me so that you're checking if I am doing it correctly? Okay. Did you do it beforehand? Okay. Al-Balo. Which of the following statements these are correct regarding blood circulation? Animals with a single circulation do not have lungs. What do you mean by a single circulation? What do you mean by a single circulation? You have a heart with only two chambers. You have a ventricle and an atrium. From the ventricle, you pump blood into gills. Always it is gills. And from the gills, the blood goes into your body cells, and then the blood comes back into the atrium. This is what we call as a single circulation. The blood goes through the heart only once during a single circulation. There's only two chambers, one atria, one ventricle. So, what did I say? I called them the gas exchange surfaces gills, because the single circulation is only found in fish, osteichthyes and chondrichthyes. Okay. Single circulation do not have lungs. Correct. Okay. Then in single circulation, blood flows from respiratory organs to other organs under reduced pressure. This is the body cells. Okay. So from when the ventricle contracts and pumps blood into the gills, from the gills to the body, how is the blood flowing? Because the gills are a capillary network. So in that capillary network, when the blood is going, pressure is going to be dropped, and then onwards it is going to be traveling at a low pressure. Okay. So that is correct, at a reduced pressure. Animals with a single circulation have two or three chambers. No, they have only two chambers. What are the two chambers? One atria, one ventricle. In double circulation, blood flows through the lungs twice during a complete circulation through the body. Can you see? If you didn't read this very carefully, you're going to take this as correct. Yes, a double circulation, blood flows through the heart twice during a complete circulation, not the lungs. Blood goes to the lungs and comes back only once. Right? That's it. Okay. So false. Myoglobin is not present in the muscles of animals having a single circulation. That is also false. Myoglobin, you can write down, is found in all chordate muscles. It's an oxygen storage molecule. Myoglobin is an oxygen storage molecule. Okay. So you can check A and B, no? Yeah. Yeah. Can you see? Another three further MCQs are from animal form. So up to 14 MCQs are from animal form. So you can see how much of a weight you have to give. Yeah, 14 MCQs. One full structured essay. Okay. Sensory receptors are connected to the nervous system. What do you think? Yes, that is one of the features that is under the characteristic of a sensor receptor. Always connected to the nervous system, right? Otherwise, how can you send their signals? Okay. Then Include specialized glands designed to receive specific stimuli. What do you think? Include specialized glands designed to receive specific stimuli. What do you think? Anyone? What are the characteristics of a sensory receptor that you know? What are the characteristics of a sensory receptor? Yeah. No, it is usually the glands don't come under uh, sensor receptors. Okay. They are either modified neurons, modified epithelial cells. Okay. Clear? So forth. So sensory adaptation. What do you mean by sensory adaptation? If you have siblings, you have sensory adaptation, or live, or if you study close to a road, if you if your room is closer to a busy road, or if you have neighbors who are noisy and you have to study, then you must have mastered sensory adaptation. That means after some time, if you are getting a repeated stimulus, you are going to be the stimulus energy is going to be reduced. You are going to start to ignore the stimulus. Okay. Yes. This is very common for hearing a loud sound. You stop paying attention to that sound. Smells, you can have visual adaptation. Sorry, sensory adaptation. Can amplify the sensory signal. What do you think? A receptor can amplify. Yes, a receptor can amplify the signal. Detect stimuli that arise only in the external environment. No, we just talked about. Where do you have aortic chemoreceptors that are found inside your body in the aorta? Then in the medullary chemoreceptors, medulla chemoreceptors. Then you have so many stretch receptors in your lungs, in your stomach, in the uterus, in the then also touch receptors are found in the nipples where the when the baby is sucking the milk is released. So many receptors are there even inside the body. Okay, so force can found in the internal environment as well. Yeah. Myoglobin stores oxygen in chordates. It's found only in chordates, myoglobin. Okay, it's found only in chordates, particularly in chordate muscles. Next, okay, Leydig cells. What do you know about Leydig cells? Yes, what do you know about Leydig cells? Secrete testosterone. Yes, Leydig cells secrete testosterone. Produce the fluid required for the transport of sperm. That is not done by any of the things that is inside. So Leydig cells are found inside the testis. No, do you does the testis produce any liquid? No, the testis only produce the sperm. Who is producing the sort of the suspensory liquid or the liquid part of the semen? Seminiferous. Sorry, seminal vesicles, prostate gland, and the tuti bulbourethral glands. Okay, they do not produce. So you can write down what are the glands that are producing the fluid: seminal vesicles, prostate, and bulbourethral glands. Okay. Nourish the cells that are in different stages of spermatogenesis. That is done by who? Nourish, again, I want you to remember the word support, right? Support. Okay. Nourish and provide support is by who? S for support. Support is by Sertoli. Support is by Sertoli. Nourish are located in the connective tissue. Yes, we actually call them connective cells. Okay, Leydig cells are actually called as connective cells. because they are found in between seminiferous tubules, in between one seminiferous tubule and the other. Very correct. Okay. Provide attachment for different cells. It is again support. Ne? Provide attachment for cells in different stages of thermogenesis. This is again support. Support is by Sertoli. Support is by Sertoli. Okay. Support is by Sertoli. Okay. Next, we'll jump to plant form questions. Take a chuti break, maybe 2-3 minutes. Chuti break क्या करेंगे? 2-3 minutes. So we'll go from question number 12 onwards. Okay. Take a chuti break, 1 or 2 minutes. Okay, shall we restart? Let's continue. Okay, question number 12 onwards. Let's go. Uh, is the following statement regarding companions? So, whenever you, so can you see what they're expecting of you in the A level paper, right? When just by giving you just one word, you are expected to recall everything what is related to what tissue, which part. Okay, so companion cells are something to do with the phloem tissue. Okay, so you know phloem tissue is something is a tissue that we consider as a living tissue. It's a tissue that we consider as a living tissue. So the main role in the phloem tissue is the sieve tube element. Right? So sieve tube elements are connected with each other, and you have what sieve plates at the each end of the sieve tube element. Okay. Then these sieve tube elements have a friend that is sort of one for one sieve tube element that is called a companion cell. So the companion cell has all the organelles that are required for the functioning of the sieve tube element. Because why the sieve tube element? The cytoplasm has been reduced to a peripheral layer. That is for the easy transport of material through the sieve tube element. Okay. Got it? Okay. So regarding companion cells, we consider the phloem tissue to be a living tissue. Right? So sieve tube elements are living, companion cells are living, then you have phloem parenchyma. Only deadly cells are your fibers, right? Phloem fibers. So of course not. They are living at maturity. They help in phloem unloading. What do you think? Phloem loading and unloading? Yes, they can have a help. They can help in phloem unloading. Okay, so that is the correct statement. They connect with adjacent cells by desmosomes. No, what do you mean by a desmosome? A desmosome is like an anchor junction. Anchor junctions are found where? Connecting muscle cells together. Anchor junctions are found connecting muscle cells together. They connect your cytoskeletons together. So even plant cells do not have a proper cytoskeleton. Okay, so what is this connection between the companion cell and the CO2 element? Those are plasmodesmata. You can write down here. They connect with the adjacent cells by plasmodesmata, not desmosomes. They are present alongside each sieve tube elements in gymnosperms and angiosperms. Sieve tube elements are only found in what? Can you find sieve tube elements in gymnosperms? No. Sieve tube elements are only found in angiosperm phloem. Okay. CO2 elements are methane you can write down only found in angiosperms. They are not found in gymnosperms. Okay, only found in angiosperms. Their cytoplasm is reduced and present as a thin layer close to the cell wall. What are they talking about here? They are talking about a sieve tube element here. They're talking about a sieve tube element here, not a companion cell. So, the question you can remember, you may forget what are they asking about in the question. They're asking about companion cells. Okay, let's move on. Leaves, okay, 13. Correct statement regarding plant leaves. Correct statement regarding plant leaves. Leaves are vertically arranged in some plants to capture light efficiency in low light conditions. So these questions are given for people, just to answer with some general knowledge. Right? If you have leaves like this, can you capture light efficiently? No, you have to have leaves horizontal. Yeah? In monocot leaves, chloroplasts are more abundant in palisade than spongy. Yes. What do you think? What do you think? Do you have more chloroplast in palisade than in spongy? When I make amatak forget this statement. In monocot leaves, forget about monocot leaves. Do you have more chloroplast in palisade than in spongy? Yes, you do. You have more chloroplasts in palisade mesophils than in spongy mesophils. But now consider with the monocot, you don't have a separation of mesophils such as palisade or spongy in monocot. Okay, so this statement is wrong. But this part, yes, of course. If you take your, if you compare your palisades are like sort of this sort of cells. Eh? They are sort of elongated, and your spongy are more sort of loosely arranged. Okay, sort of inner to the cell. Palisades are tightly packed with chloroplasts. Palisades are tightly packed with chloroplasts. Spongy do not have as much as chloroplasts as palisade. But in monocot, there is no differentiation. Everyone is just mesophyll cells. Angiosperms can be identified due to the net-like venation. This is called reticulate venation. Reticulate venation. Is it found in so okay. What specific angiosperms? Angiosperms are of two types. And flowering plants are of two types. What are the two types? Monocots and dicots. Monocots and dicots. So, can anyone tell me who has net-like venation? Who has net-like venation? Is it dicots or monocots? Is it dicots or monocots? Dicot. So, angiosperms can be identified due to net-like venation. What do you think? Not really, no? Because you can have, what is the other venation? What is the other venation that you can have? When we have polcolor coconut leaves, you have all the veins running in the same direction. That is called parallel venation. So, angiosperms can have parallel venation as well. So, you can't use it as identifying factor. What is the best way to identify angiosperm? If there is a flower. Arrangement of leaves on the stem is called leaf orientation. What do you think? Arrangement of leaves on the stem, is it called leaf orientation? What do you call that? Arrangement of leaves on the stem. What do you call that? Ken, there's a nice word for that. Phyllotaxy. Okay. Phyllo sort of refers to leaf. Okay. Sporophylls. Yeah. Again. Microphylls, megaphylls, phylls usually are used to call leaves. So phyllotaxy is the arrangement of leaves. Okay. Plant inhabiting very cold environments bear the smallest leaves. Yes, if you are living in a very cold environment, your leaves are likely to be damaged by the snow or the very cold. So you can have small leaves. Correct. Common general knowledge. No? Okay, moving on. Question number 14. Any questions up till now? Some events that occur at the source during phloem translocation of angiosperms according to the pressure flow hypothesis are as follows. Okay, pressure flow hypothesis. How does it work? How does the pressure flow hypothesis work? Okay, I'll quickly explain the pressure flow hypothesis. So, how does the pressure flow hypothesis work? Of course, it occurs through the phloem. No? Okay, it occurs through the phloem tissue. Pressure flow hypothesis is the hypothesis that we use to explain how phloem is transported. Okay, so you have a place where phloem is loaded into the phloem tissue. Let's take this the phloem loading place as a leaf. So we call that a source. Then let's take as the phloem unloading place as maybe a route, a storage route. Okay, where right, a storage route. Okay, then so what is going to happen when you load sugar into this phloem cu tube element at the source, what's going to happen? The water potential is going to reduce. When the water potential reduces, who is going very nearby usually to a phloem? There's a xylem. There's a xylem tissue going very closer to it. What's going to happen is due to the reduced water potential, water is going to come from xylem to the phloem. Okay. Then what's going to happen? The pressure in the phloem near the source increases. That is going to generate a positive pressure that is going to push this phloem sap towards the what we call this the sink, where the sugar is used for metabolism or converted to insoluble for product form such as polysaccharides, starch again. Okay. So when you arrive at the sink, what's going to happen? The sugar will be unloaded. Sugar will be unloaded. Therefore, when the sugar is unloaded, the water potential of the sink inside the phloem increases. Therefore, what's going to happen? The water will come back into the cycle. So now can you see the pressure decreases? So there's a pressure gradient between the source and the sink due to the differences in the pressure. There's a pressure gradient. So therefore, that's why we call this pressure flow hypothesis. Pressure flow hypothesis. Okay. So what is the sequence of events? What is the sequence of events? Right, ABC. Flow of water into the CU tube from xylem. I think something, okay. Generation of positive pressure inside the CU tube. So we are asking, okay, right. Reduction of water potential inside the CU tube. I think something that is missing here, අපි මුලින්ම මෙතනට එකක් add කරමු. Sugar loading is the first thing to happen. Okay, sugar loading happens. Then afterwards, what should happen? Sugar loading happens. Then afterwards, water potential inside the CU tube reduces. So that is the next thing to happen. Then when you reduce the water potential, what's going to happen? Water comes into the CU tube from the xylem, number 2. That is going to increase the pressure, generating a positive pressure. So CAB, answer number 5. $15. 15, what is the answer? Atmospheric air is the only source of which of the following elements required by plants? So plants are what? Plants are photoautotrophs. Photo means their energy source is by light. Auto means they fix inorganic carbon. What is the inorganic carbon source that is available to them? Carbon dioxide. Because they are photoautotrophs, they should use atmospheric carbon. So, what is their only carbon source? I mean, the atmospheric air. So, carbon is the only source for carbon dioxide. I'm sorry, atmospheric carbon dioxide is the only carbon source. Okay, let's continue. 16. Question number 1. 16. Yes, some characteristics of two species of plants are given below. A, dominant sporophyte, okay. Gametophyte is reduced, sporophyte and gametophyte are photosynthetic and independent. As soon as you hear the word, okay, the sporophyte is dominant, that means you are in a vascular plant section, no? Because only non-vascular plants such as your pogonatum or a bryophyte is going to have a dominant gametophyte. Yes. So sporophyte is dominant and bryophytes are out. So you can this pogonatum wage, all the answers are gone. Then gametophyte is reduced, okay? Sporophyte and gametophyte are both photosynthetic and independent. There is only one species where you have an independent gametophyte that is also reduced. That is the heart-shaped gametophyte. Remember, who is the heart-shaped gametophyte? That is in your can. Who has a heart-shaped gametophyte? Nephrolepis. Nephrolepis gametophyte. So you have two answers with nephrolepis. So who is the second one? Phosphite is dominant photosynthetic, gametophyte is reduced and partially dependent. Partially dependent sporophyte. You can have a partially dependent sporophyte that is having some stored food, but also having a bit of photosynthetic tissue. Who is that? That is the Selaginella. Actually, this is actually the female gametophyte of the Selaginella. Okay, the female gametophyte of the Selaginella. But you can take answer number 1. Can you understand how I arrived at the answer? Because there's only one with the independent gametophyte that is not a non-vascular, that is Nephrolepis. The heart-shaped green color thallus, macroscopic thallus. Remember? Okay, final plant form question. Oh, there's a question. Semen is solely consists of the secretions of the three accessory glands. Yes. Oh, wait. Semen. Semen ಗೆಲ್ಲ ಏನು? So, I think that's the problem. Semen is equal to sperm plus secretions of the accessory glands. Then semen without sperm is like, I don't know, tea without the tea made without the tea leaves, adding tea leaves. So you have added the sugar and the milk, but you call it a tea, ne? No, you can't call it a tea. So semen has sperms. Then you have the fluid that is to provide nourishment and other purposes. Okay. So semen is composed of sperms and the secretions of the three glands. Anyway, 17. Which of the following hormones in plants is stimulated by water deficit? Okay, this is direct MCQ. I mean, direct theory application. Abscisic acid is the plant growth substance that is released in response to drought conditions, water deficiency. Okay, so we have how many? 5 plant form questions. Can you understand? Out of the 55. Okay, big amount. From plant form. Of course, you know there's going to be maybe 2/3 of a structure, they say. There's nothing from in this section. There's nothing from plant form. Neither. Ah, yeah, there is. Maybe question number 42. What does it say? Primary growth of roots. During the primary growth of roots, root apical meristem produces new cells to the both sides. What do you think? Can you recall the root apical meristem? The root apical meristem may have pointed. Okay, so this is the root apical meristem. Dense cytoplasm, spherical cells. Okay, so it produces cells to the inside. Those cells are going to undergo elongation and then they are going to undergo differentiation. So, maybe root hairs, they become root hairs, they become xylem and phloem, right? So, that's going to happen to the cells produced inside. What is going to happen to the cells that is produced to the outside? They become like the helmet. That is the root cap. Correct. You can say out to the outside root cap to the inside, they differentiate, elongate and differentiate. Methane cells produced outward by the root apical meristem form the root cap. Vascular tissues are produced by vascular cambium. Vascular tissues are produced by vascular cambium. What do you think? හොඳට කියවන්න. So එකපාරටම if someone gave you just this statement number 3, vascular tissues are produced by vascular cambium, forget the stem එක අමතක කරන්න. Forget about the question. If someone gave you vascular tissues are produced by vascular cambium, is it wrong? Is it wrong? හොඳට හිතන්න. Then logically analyze කරන්න. These are the past paper questions. You have to go to the exam after nice analysis. Is it wrong to say vascular cambium produces vascular tissues? No, the namimathina, it's vascular cambium. But the question is asking about primary growth. So what are the vascular tissues produced by vascular cambium? Yes, Kian, xylem, phloem. But is it just xylem and phloem? Can anyone tell me? It's what are the xylems and phloems produced by the vascular cambium? It is secondary xylem. Secondary xylem and secondary phloem are produced by vascular cambium. Vascular cambium is a secondary meristematic tissue. That's why this is wrong, because they are asking primary growth. Some cells produce outward by the root apical meristem, elongate and push the root. No, if you are producing cells outwards, that is going to become the root cap. If you are producing cells inwards, then if you can correct this to say produce inwards, elongate and push the root. E. Epidermis splits due to being pushed outwards. That happen during again primary growth? If you gave it without the statement, that is correct. During the secondary growth, yes, in the secondary growth, when the inner tissues are getting formed, like secondary xylem, secondary phloem, cork tissue, when it is being produced, the epidermis is going to be pushed and crack and fall away. But does it happen during primary growth? No. Okay, so about 6 MCQs are from plant form. Okay, what else do you want to do? Which questions do you want me to do next? So we have covered almost 25 questions just from 2 lessons. Okay, we'll do a bit of evolution. Okay, so evolution starts from from question number 7 onwards. Okay, we'll do some evolution. Okay, let's continue. Right. Which of the following statements are important in explaining Darwin Wallace Tutikalagarbu evolution? Okay, but we have to, these are marks bringing lessons. Okay, easy marks. Most important in explaining. Organisms acquire suitable adaptations according to the needs of the environment during their lifetime. That is something from what? Yes, that is inheritance of acquired characteristics. Not something done explained by Darwin's theory. Yes, so this is more like Lamarck. This is more like the Lamarck's theory. That is the first part of Lamarck's theory. No? Second one, adaptations acquired during the lifetime are passed to the next generation. That is again Lamarck. Inheritance of acquired characteristics. Okay. The theory of use and disuse and inheritance of acquired characteristics. Okay. Second one, sorry, third one, favorable characters are passed to the offspring through genetic factors. Okay. Is this Darwin Wallace? Very important. Then, if you had only a small idea or didn't have a great idea about the theories of evolution, this sounds good, ne? Characters are passed to the offspring. But is it Darwin Wallace theory or is it something else? Anyone? Any idea? Is it Darwin Wallace theory? Yeah? No. This is the modern version of the Darwin. So upgraded Darwin's version because Darwin didn't know about genetic factors. So this is neo-Darwinism or modern Darwinism. Darwin didn't know how to explain the way that genetic, whatever the characters you acquired, how is it passed to the next generation? He didn't know that because Mendel came later. Gregor Mendel came later explaining about how there are inherited factors. 4. Each species produce more offspring than the environment can accommodate. Yes, that is part of Darwin Wallace theory. So tonight itself, I want you to finish studying just within 10 minutes. Within 10 minutes, finish the final study of theories of evolution. How the theories of evolution? Just final study. Okay. Uh, There it can be question in the structured essay. It can come as a, short note, right? We can never know. Okay. So those are actually observations. This is an observation of Darwin. Adaptations result in changes in genetic material. Again, Darwin didn't know about genetic material, so you can't take that. Darwin didn't know anything about genes. 8. Three genera with circular chromosomes, histone associated with DNA, and several kinds of RNA polymerases. So you are supposed to have circular chromosomes. That means, of course, you are either, if you are having circular chromosomes, if you are either a domain bacteria or archaea. Neither. But if you have histones associated with DNA, then you can cut bacteria. Because who only has histones associated with DNA? Out of these two, archaea, eukarya also have. Okay? Because in some species of archaea, you can have histones associated with DNA, even though they have circular chromosomes. Several kinds of RNA polymerases. Hold on. They are talking about a list. No, I'm sorry. Okay. So three genera with chromosomes, circular chromosomes. Yes, sorry, you can't. Yeah. Circular chromosomes are associated, found in bacteria and archaea. Okay. Then histones associated with DNA is found in archaea and eukaryote. Several kinds of RNA polymerases also found in archaea and eukaryote. So, what are the examples? Thermococcus, methanococcus, those are all archaebacteria. Halobacteria, all archaebacteria. Anabina, Pseudomonas, those are all bacteria. So, all the answers for one is correct. For two, you need to have either a eukaryote or archaebacteria. So, amoeba correct, halobacteria, salmonella you can't take, cycus you can't take, anabina you can't take. Anabina is a bacteria. It's actually a cyanobacteria. Ne? Anabina is a cyanobacteria. Then, finally, what can you take? Methanococcus you can take, Nitrosomonas you can't take, Nostoc you can't take. So, it's the answer. Answer number 1. Can you understand how I got this? Circular chromosomes are found in both bacteria and archaea. Histones associated with DNA found in archaea and eukarya. Several kinds of RNA polymerases also in archaea and eukarya. So you have to go with the cancellation method. The answers kapaගෙන යන්න වෙනවා. So Thermococcus, Methanococcus, Anabina ඔක්කොම is matching because either bacteria archaea, Halobacteria, Methanococcus, Thermococcus are all archaebacteria. Okay. Then Anabina and Pseudomonas. Anabina is a cyanobacterium. Pseudomonas is a bacteria. Nostoc is again a cyanobacteria. Okay. Salmonella is a bacteria, so you can't take it for the second one. Histones associated DNA. So can you understand how we got the answer? Okay. Next question. 9. Okay. Seedless vascular plants are evolutionary closer to hornworts than mosses. What do you think? Yes, that is true. Seedless vascular plants are actually closer to hornworts. Okay, that is directly stated. Okay, who are your hornworts? You can get this answer from that evolution chart in page number 87 of the resource book. Can you see? So you have liverworts as the oldest, then you have mosses, then you have hornworts, then afterwards you have your lycophyta. So you start the seedless vascular plants. So who is evolutionary closest? Hornworts are evolutionary closest to lycophytes. Lycophytes are what? Seedless vascular plants. Seedless vascular plants bear spores, yes, but they do not have any relation. Then on chemistry paper you have the question number 40 to 50 right? Explained by B, right? So they bear spores because they don't have seeds, seedless vascular plants. So it is nothing to do with evolutionary association. Understood? Clear? Okay. Next question. 10. 4 structures present in protists are as follows. 10. Okay. Organisms having A, B, C, and D are respectively. So you have a multicellular thallus. So multicellular thallus, now it has to be some sort of algae. It has to be some sort of algae because Euglena, Paramecium, your protozoa are all unicellular. Contractile vacuoles. Okay, you can have several options. Pellicle. Pellicle can be found in several protozoa. Okay, pellicle can be found in several protozoa, such as your Euglena and Paramecium both. Okay, then contractile vacuole can be found in Euglena and Paramecium as well as amoeba, all three. Cell walls can be found in usually all algae. So you need to have an algae. So sargassum, ulva, gelidium, ovitica, for the answer for the first answer, okay. Contractile vacuole, diatom you can't take, Euglena you can take, amoeba you can take, paramecium you can take, Euglena you can take. So first answer we can cut off already. Then C, pellicle is found in paramecium, not in ulva, not in amoeba, not in sargassum. So we have arrived at the answer. Answer number 2. Can you understand? So if it's multicellular, it has to be an algae out of the proteins. What are the two types of proteins? You have algae and protozoa. And multicellular, it's algae. Contractile vacuoles are usually found in all three. What are the all three types of protozoa that you know? Euglena, Paramecium, and amoeba. All three you have. Pellicle is found in Euglena and Paramecium only. Cell wall you can be found in all algae, Gelidium, Sargassum, diatoms. Okay. All right. We'll do this question and stop for the day. Okay. We'll discuss the remaining microbiology and molecular questions. Okay. Coming week. Which of the following indicate two features found in organisms of the same phylum? Okay. Two features found in the same phylum. Okay. Question number 11. What do you think? Heart absent, endoskeleton present. Heart absent, endoskeleton present. Who has a skeleton but without a heart? Heartless man with a endoskeleton. Who is this? Having bony plates. Sorry, calcium carbonate plates. Who's having calcium carbonate plates as their skeleton? Ken? Echinodermates. Echinodermates. They don't have a heart. They have a water vascular system. Okay. B. Heart absent, jointed legs present. Who doesn't have a heart? But without, I mean, usually they have, no? Because arthropods, they have jointed legs are of course found in arthropods. But this heart absent, it's false, no? Because they usually have a open circulatory system. You can write here, they usually have open circulatory system, right? But you can't say heart absent. Anus absent, tentacles present around the mouth. What do you think? Sorry. Anus absent and tentacles. Tentacles are found in cnidarians, such as your sea anemones. Okay. They don't have an anus, no? Because they have mouth and the anus, the same opening. They have only one opening. So you can't take C. Okay. I'm sorry, you can take C. Sorry. Anus absent means yes, it is present in cnidarians, tentacles. Anus absent show asexual reproduction. Anus absent show asexual reproduction. For this also, you can take cnidaria, no? Because if you take an cnidarian like hydra, if you take you can write down here. If you take an cnidarian like hydra, they have the asexual reproduction method of. Can anyone tell me what is the reproduction method of hydra? What is the reproduction method of hydra? Anyone? Yes. Budding, fragmentation and budding. So cnidaria you can take. So except B, everything is correct. No, can you understand why I couldn't take B? Because if you are having jointed legs, you are an arthropod. But if you are an arthropod, you are going to have open circulatory system. For that, you need a heart. So ACD. Okay, right. So this is the plan that we are going to go ahead. Join next Wednesday with the final day of applied biology, and there we are going to finish the applied and also all the past paper questions that have been there. So I'm not going to do the applied biology questions here because they will be covered the next week at the revision class. Okay, and yeah, so that is going to be the end of the revision class. Then we'll have some molecular questions from the past papers in the revision class as a discussion. Then we'll do the 2022 paper, 23 paper. Okay, then that is the end of the paper class as well. So join with the paper class next week as well. Okay, new students, you can watch the last week recording at the website. Okay, so this is the final phase that you are going. So stay motivated but don't get stressed. Okay, keep a nice balance. Okay, you still have time, a lot of time to work, right? Exactly 30 days are remaining. Okay, you can do a lot, particularly for bio. You can do a lot because the last minute things that you take into your brain are going to be able to be taken out in the exam. Antimote. So the main thing is you don't lose. I mean, you become, because of the stress, you don't waste time. That is the main thing. Okay, so keep that in mind. Keep in mind that staying calm and focused is going to get you the result, not being stressed. Okay, so intentionally think about that. Don't become stressed. Just keep working. Okay. සුට අර බය වෙන්න එපා. බය වෙලා ඉන්න එකට බය වෙන්න එපා කිව්වේ නැතුව වැඩක් නෑ තමයි. But it's the logical thing to do. So do the logical thing. Okay. Hari, okay, we'll finish for the day.